In this lesson, we'll learn about how mutations affect the proteins that they code for. One important distinction to make is that there's a difference between somatic and germline mutations. Somatic mutations involve changes to the DNA in non-reproductive cells. As a result, any cell that originates from the original mutant cell will have that mutation. However, somatic mutations are never passed on to offspring because they don't occur in the egg or sperm cells that become the next generation. Somatic mutations may be the result of errors in copying DNA during S phase of the cell cycle, or they may be the result of something in the environment. Radiation, including UV rays from the sun, can cause mutations, as can exposure to a number of chemicals called mutagens. Remember from previous lessons that mutations may affect small regions of DNA, or they may affect the structure of entire chromosomes. Germline mutations, however, may be passed on to the individual's offspring with a 50% chance. If the offspring does inherit a mutation, they may be a heterozygote or, depending on the other parent, a homozygote. In any case, every single cell of the offspring will have that mutation. Germline mutations are most likely to be a result of copying DNA during S phase before meiosis, but it's also possible, though much less likely, to be due to environmental factors. So let's take a look at the different types of mutations and their consequences. A point mutation is a mutation that affects only one or a few nucleotides. All of the mutations we're looking at in this lesson are point mutations, as we've looked at larger scale mutations and chromosome rearrangements previously. This example is a mutation that causes sickle cell anemia. Worldwide, most individuals have this wild-type beta-globin allele. Although the gene is much longer, the important code here is the one with a CTC on the template strand and GAG on the coding strand. The mRNA that's produced is the same as on the coding strand, that is, GAG. If we look up the codon GAG in the codon table that we've seen previously, we find that the GAG codes for glutamic acid. Remember that it codes for glutamic acid because the transfer RNA with the anticodon CUC, that is complementary to GAG, carries the glutamic acid residue. Some people inherit a mutation in this gene in which the middle base of this codon on the coding strand is a thymine instead of an adenine. As a result, the mRNA is GUG, and this codon codes for valine instead of glutamic acid. What's the consequence of this change? Well, this mutation causes the hemoglobin to bind together in a way that changes the shape of red blood cells when the blood cells are short of oxygen. This shape change causes the cells to be much longer than normal and less flexible, causing them to get stuck in the smallest capillaries. Historically, individuals that are homozygous for this allele usually died as infants or as young children. Heterozygotes, however, may suffer some effects of sickle cell anemia, but they actually have an advantage over normal homozygotes because they're resistant to the pathogen that causes malaria and is spread by mosquitoes. You can learn more about this mutation, which affects people in equatorial Africa and Asia, in the link above. Not all mutations have such a dramatic effect. In fact, sometimes there's no effect at all at the phenotypic or even the molecular level. In this example, the fourth codon is GGC, and it codes for the amino acid glycine. If a mutation caused the template strand to change to ACC, and the coding strand therefore to be GGT, the mRNA codon would then be GGU, which still codes for glycine. In this case, there was a mutation in the DNA, but there was no effect of the mutation as long as the resulting protein is still exactly the same. Since the protein is the same, there will be no effect on the phenotype of the individual. This is called a silent mutation because there are no phenotypic effects, even at the protein level. Okay, let's look at a handful of mutations of different types. Here, I've given a DNA sequence listing both the coding and template strands. Remember that the template strand is the strand that is used for synthesizing the mRNA, but the coding strand has almost the same sequence as the mRNA because both the coding strand and the mRNA are complementary to the same template strand. When you're looking at a problem like this, you need to pay special attention to the strand orientation. Remember that mRNA, like DNA, is always synthesized from the 3' to 5' end. Also, when you're trying to figure out the sequence of mRNA or protein, try to do it like the cell does it. That is, transcribe the entire sequence unless you know where the transcription start site is. Transcribe all the way to the end of the sequence, then find the start codon. The start codon determines the reading frame. We'll come back to that, the reading frames, in a moment, but in this sequence here, I've transcribed the DNA into mRNA, being careful to get the orientation correct. Once you've found the start codon, mark out the codons of the reading frame 3 at a time, starting from the start codon. Once you've done that, use the codons and the codon table to translate into protein. Remember that the codon table is for mRNA, as we've used it for here. This is easy to remember as there are uracil instead of thymine in the codon table. In this polypeptide, there are 6 amino acids. Now, we do count the methionine because it's an amino acid, but we don't count the stop codon because no amino acid is added as a result of that codon. In this lower sequence here, there's been a mutation that replaces the G of the coding strand with an A at base number 9. After you transcribe and translate this sequence, you would see that the 3rd amino acid lysine was replaced by an arginine. This is a missense mutation because the amino acid changes. This is identical to the situation with sickle cell anemia. In this case, like previously, there's been a single base change, also at base number 9. However, this change results in the glycine being replaced by a stop codon. This is known as a nonsense mutation, where an early stop codon is introduced and the polypeptide is not full length. We mentioned earlier that the start codon sets the reading frame. Consider the DNA sequence in the middle of this diagram. On top, you have the 5' to 3' strand, and below that, you have its complement. If you consider that the first 3 bases constitute a codon, then this would set the reading frame to be frame 3. In this reading frame, bases 1, 2, and 3 are a codon, bases 4, 5, and 6 are a codon, and bases 7, 8, and 9 are a codon, etc. If, on the other hand, bases 2, 3, and 4 constitute the first reading frame, then we have reading frame 2 here. Similarly, if bases 3, 4, and 5 are the first codon, then we have frame 1. The same thing is true for the other strand in the case that that strand is the coding strand, except that we have to read in reverse along that strand. Thus, for any strand of DNA, there are 6 possible reading frames. Researchers sometimes look for what is known as an open reading frame when they're trying to figure out where a gene starts and ends. They reason that a gene has a long distance between the start and the first stop codon. In this simple example, frame 3 would not be an open reading frame because it has the start but no stop codon. Frame 2 would also not be an open reading frame because there's no start codon, and the same is true for frames 4 and 5. Frame 1, however, has both a start codon and, at a good distance, a stop codon. If this distance was great enough to accommodate a gene, it would be considered an open reading frame. The last class of mutations are insertions and deletions, and are often called indels. Insertions and deletions cause frameshift mutations, so-called because they change the reading frame after it has been set by the start codon. In this example, we're just simply using the English sentence, "The fat cat ate the rat." This is a useful sentence because each word is exactly 3 letters, just like mRNA codons. If during DNA replication an A is inserted into the word cat, then the reading frame is off. The ribosome maintains the reading frame that it set at the start codon, but now those reading frames after the inserted base specify for very different amino acids, in the same way that this sentence no longer makes any sense. Here's an example with actual DNA. In this case, a T was inserted on the coding strand at base 11 and pushed all the other bases down 1. When the ribosome comes to translate this, it will maintain the reading frame set at the start codon. Therefore, all codons after the insertion will specify different amino acids. Note that all of the green amino acids are now different, and importantly, there is no stop codon. It's often the case that an insertion like this will often introduce a stop codon prematurely, causing the translation to end with a shortened polypeptide. There's also a very similar effect if a base is deleted instead of added. In this case, an A was deleted, causing all downstream codons to be frameshifted. Note, none of the amino acids that are added to the polypeptide will be correct. Sometimes, however, insertions and deletions happen in multiples of 3 bases. When this happens, a frameshift does not occur, and the mRNA maintains essentially the same codons, resulting in a polypeptide that has fewer or more amino acids, but is almost the same as the unmutated allele. Using the English example, you can see that the sentence may be changed to "The big fat cat ate the rat," or "The big sly fat cat ate the rat," or with a deletion, simply "The cat ate the rat." Before we end, let's take a minute to think about what the phenotypic effects of these mutations may be, and whether they can be predicted. Usually, we can't predict the effect on the phenotype because the interaction of a protein to produce a particular phenotype is very complex. It's usually much easier to predict what the effects of a mutation are on the function of a protein. At one end of the spectrum, a mutation may have no effect on protein function, and at the other end, it may cause the protein to be completely non-functional. Between these two extremes, there's a range of effects. For example, sickle cell trait causes the protein to be less functional, especially under oxygen stress, but it is always partially functional. Not surprisingly, silent mutations have no effect on the protein function because the protein is exactly the same as the wild-type allele. Missense mutations, which change a single amino acid, often have only small negative effects. If the amino acid is deeply embedded in a protein in an inconsequential site, or the replacement amino acid has similar biochemical characteristics, then the effect of the protein function is likely to be small. However, there may be a large effect on protein function, such as if the mutation results in a change in the enzyme's binding site. In general, the variable effects of missense mutations are very difficult to predict. The negative effects of nonsense and frameshift mutations are generally large. This is because in both nonsense and frameshift mutations, large portions of the polypeptide are either missing or the polypeptide is just totally different. The exception to this may be when such a mutation occurs very close to the C terminus of the polypeptide chain, resulting in a polypeptide that's very similar to the normal allele. Finally, in-frame insertions and in-frame deletions usually have limited negative consequences for the protein function. This is because the polypeptide remains largely the same. As with missense mutations, if the mutation involves an active site or some other important part of the polypeptide, the consequence of mutation may be more severe.